Hello, everyone. We're just giving everyone a moment to enter the room and we'll begin shortly. Okay, my distinguished panel, are you ready to begin? Okay. Greetings, everyone. From the American Physiological Society, I'm Patricia Terrell, the Meetings and Program Coordinator for APS and moderator for today's webinar. Thank you for taking the time from your busy schedules to attend this very unique and exciting presentation titled A Novel 3D High Throughput Co-Culture System for Multiple Myeloma. Before we begin, here are a few important housekeeping notes. The webinar is being recorded and made available for on-demand viewing following the event. To maintain scientific integrity and respect for our presenters, please refrain from taking photos or videos of this presentation in whole or in part. Now, if you look at the bottom of your Zoom screen, you will find the Q&A and chat features. To ensure the prompt delivery and receipt of your questions, we ask that you use only the Q&A feature. And you may do so at any time during the presentation. We will answer all of your questions immediately following the presentation. In addition, live captions are available by clicking the live transcript icon, also at the bottom of your Zoom screen. So without further ado, let's get this webinar started. It is my true pleasure to introduce this incredible team of scientists and businessmen. Dr. Andreas Thomsen is a radiation oncology specialist at the University of Freiburg, Germany. And Dr. Marco Loh is the Chief Operating Officer at ABC Bioply in Solothurn, Switzerland. So what I would like to do now is turn the presentation over to you, Dr. Thomsen. You may begin.
So I need to have permission to share my screen. At the moment, I cannot do this yet. Wonderful. Yes, Patricia, thank you so much for this nice introduction. And hello, everybody. As an introduction, I want to just say that we need to disintegrate nature, we need to disintegrate physiology to build up models, but we definitely must not oversimplify. So we cannot use monolayer cultures as a perfect model for biology, because biology is complex, because biology is not flat, but it's crowded. You see that in the small tumor region or in the micrometastasis, we have a huge crowding of cells. All these cells build up gradients, they communicate with the neighbor cells, and we need to model this to approximate reality. We must say that such a crowded condition cannot be achieved in all cases with the standard cell culture conditions, 3D cell culture conditions. So in the hanging drop or in the liquid overlay system, we have only a loose aggregation of the cells in many cases. But in the 3D CoSeedis system, where we have conical micro cavities in agarose gel, we achieve such a high density, and we have the conditions which build up gradients and will be able to model nature much more closer than any other culture system. To give you an idea how this setup is working, so we have a chip which is inserted into culture plates, and this chip has been manufactured with a high precision and with a high number of standardized microcavities. So cells are seeded by a single step, by single pipetting, and after cell aggregates have formed, we can add radiotherapy, we can add drug treatment, we can add different types of other conditions, and then we have the time to react after a number of days to weeks to do evaluation. So we can simply measure volume of these cell aggregates, which is possible by this conical geometry. We could harvest the cell aggregates and do reseeding into extracellular matrix gel, or harvest proteins or DNA, RNA, whatever. We can do imaging, and we even can do histology, which is a very nice opportunity to characterize these cell aggregates which have been formed. And finally, I want to point to something which is rather new, which is relevant for all of us, because up to now, animal models were mandatory for market approval of new drugs, but the new act of the FDA law has included cell-based assays and more complex conditions apart from animal models, which can be used to show the safety and effectiveness of a drug candidate. So thank you so much for this first views, and I hand over to my colleagues.
Thank you very much, Andreas. My name is Marco Loy. I'm the CEO of ABC Bioapply. Now, after Dr. Thomsen's excellent introduction of the 3D CoSeedis technology, and before I will hand over to Dr. Waldschmidt and his implementation of 3D CoSeedis in drug testing on multiple myeloma, I would like to briefly introduce you to ABC Bioapply services and assays. ABC Bioapply offers the 3D CoSeedis technology as an integral part of its service packages to support customers in the design and analysis of physiologically relevant preclinical 3D disease models. Our value proposition depicted on this slide here starts with the definition of a custom-tailored preclinical study program. This may include the design of the appropriate disease model and the establishment of culture conditions to reproducibly make the model accessible in subsequent drug testing setups. Furthermore, it includes the definition and eventually the development of customized assay packages to investigate the previously designed 3D models. We currently offer a broad range of various assays covering pharmacodynamics, pharmacokinetics, and many more. The assay may also be an integral part of a high content analysis setup, for example, or it may consist of highly specific histological analysis. An important aspect is also that with 3D CoSeedis, ABC Bioapply is capable to provide highly accurate and physiologically relevant data on off-target toxicology. Of course, all our services will be assembled in an appropriate data report at the end of the test series. All assays developed by ABC Bioapply can also be in-licensed to be integrated in work processes at our customers' facilities. We have already existing simple assays off the shelf, so to speak, but also provide entire comprehensive analysis packages that may include several types of different assays in combination, such as the off-target toxicology testing assays or the 3D genotoxicity testing. Furthermore, we may also develop customized assays purely for the subsequent integration at the customer site. ABC Bioapply therefore can really help you to develop a specific custom-designed assay that matches your expectations. Since ABC Bioapply is the proprietary owner of the 3D CoSeedis technology, we also have the possibility to adapt the technology platform itself to suit our customers' requirements. This distinguishes ABC Bioapply as a unique player in the CRO sector and allows us to provide truly custom-designed products. Now, should you have any questions related to this webinar and ABC Bioapply services and assays coming up during the next few days, that you would like to discuss in further details with me, please feel free to contact me anytime. And that's why I put my contact details here. And with this, I would like to hand over to Patricia again, who will introduce you to Dr. Johannes Waldschmidt and his fascinating work on multiple myeloma. Thank you for the moment.
Absolutely. Thank you so much, Drs. Thomsen and Lowe. What I'd like to do is, just thank you for setting the stage to introduce this exciting scientific technology. And before we introduce Dr. Waldschmidt, we'd like to get a quick poll from our viewers to find out how many of you are currently using 3D cell culture. So let's take a quick poll and then we'll get started with the presentation. Do you currently use 3D cell culture in your research? Oh my goodness, look at the responses. %. Can I get Marco and Andreas to rejoin me?
Yep.
How do you feel about these numbers? Do they surprise you at all? 42% yes and 58% no.
Mm-hmm.
Well, actually, I'm not so much surprised. No, I mean, a lot of work is still done in 2D, but there is, however, a strong tendency to shift to three-dimensional models, particularly in preclinical test setups. Experience has shown that this is, of course, of importance since 3D models generally deliver just more reliable results. And that's what we will hear more about also later on now.
Mm-hmm.
Great.
I completely agree. Just a couple of years ago, there would have been like 10 or 15% people doing 3D culture and all the rest doing monolayers. So we
Awesome.
have a great development.
Okay, great. Well, thank you for your feedback and thank you everyone for participating in the poll. Now we're going to go ahead and get started with our presentation. And at this time, I would like to introduce Dr. Johannes Waldschmidt, who studied medicine at the University of Freiburg, Germany, the University of Uppsala in Sweden, and the University of Zurich, Switzerland. He was trained as a hematologist-oncologist at the Freiburg University Medical Center, where his experimental work has focused on characterizing the bone marrow microenvironment in multiple myeloma. As a postdoctoral fellow in the lab of Dr. Jens Lohr at the Dana-Farber Cancer Institute in Boston, Massachusetts, Dr. Waldschmidt studied cancer genomics, clonal evolution, and the development of drug resistance in multiple myeloma. He is a recipient of the 2019 Young Investigator Award by the International Myeloma Society. And in 2021, he returned to Germany to pursue his translational research in plasma cell diseases. And so before we begin the presentation, I'd like to remind you once again to use the Q&A feature to submit your questions at any time during the presentation. And at this time, we would like to present Dr. Johannes Waldschmidt.
Good afternoon, everybody from Germany. I have the great honor to speak to you today on behalf of our group at Freiburg University Medical Center. So first, thanks to everybody at APS for the opportunity to present our work in today's webinar. I will present our data along with Andreas Thomsen. We're both MDs with a special interest in vitro modeling in cancer, and Andreas will be available for all questions you might have at the end of this talk. We will present data on CoSeedis, which is a 3D high throughput co-culture system initially developed for the ex vivo cultivation of solid tumors, but we could also recently show that it has quite some potential in the arena of multiple myeloma, and this is the data I brought you today. Myeloma, which I'm sure you all know, is a plasma cell malignancy with an estimated incidence of 32,000 new cases in the US each year. And treatment of this disease has rapidly advanced over the last decade. Multiple agents have come to the clinic, but although long-term survival is now noted in quite a substantial amount of patients, the emergence of therapy-resistant disease still prevents cures. And with better survival, longer outcomes, we also have more relapses, and this is why there's still an unmet need for more individualized approaches and real-time prediction of the clinical efficacy for patients with this challenging disease in respect to certain agents that are out there on the market. The primary residing site of multiple myeloma is the bone marrow, and it has been shown by us and others that the cell-cell interaction and the support by cytokines within the bone marrow niche is fundamental to both the proliferation and drug resistance of myeloma cells. These biological premises, however, may not be fully reflected by the currently available preclinical models for myeloma, and there's actually one interesting work showing that most models remain inefficient in predicting later clinical efficacy. This is a retrospective study conducted by the myeloma clinic at Mayo Clinic, Scottsdale, Arizona, and between 1961 and 2013, 129 agents with preclinical single-agent activity against myeloma have been brought to the clinic and have been tested as single agents. However, the overall response rate for these single agents in the clinic was very disappointing, and in fact, only 10 of these agents could be validated in phase 3 trials and are nowadays approved for the treatment of multiple myeloma. So, understanding the preclinical modeling of myeloma seems really crucial to optimize the translation of in vitro findings from bench to bedside. As our knowledge on the biology of myeloma has dramatically increased over the last decade, more complex in vitro models have been introduced, thereby providing a wider range of test systems from low to high throughput testing systems. These platforms include 2D monolayers, hanging drop models, bioreactors, scaffold-based models, liquid overlay cultures, and all of them have certain advantages and certain disadvantages. 2D models allow for high throughput screening but do not adequately assess the interaction of myeloma cells with their environment. More sophisticated 3D co-culture systems have integrated additional components which permit the evaluation of a broader range of therapeutic agents and also strategies, but they increase the complexity and thereby limit the applicability of 3D models to smaller throughput series, also with some limits in reproducibility. And you can see all these different models summarized in the table shown on this slide. In the next couple of minutes, I want to present data on a novel conical agarose-based 3D microwell platform for the reproducible cultivation of primary myeloma cells ex vivo. And for the manufacturing process, we designed microwells machined into a polyoxymethylene block using computer-aided design. The resulted POM master structure had a microwell periodicity of 0.51 or 2 millimeters, and the molds were generated using PDMS loaded with 2.4 agarose, followed by centrifugation to remove the bubbles. Finally, sterilization using UV irradiation, and this actually gave us a disc-shaped microwell arrays that could fit in either 6-well or 24-well plates. And each of these microwells was actually designed as a truncated right circular cone, and this gives us certain advantages. First, other than cylindrical microwells, the conical shape centers the seeded cells, and this leads to a formation of a single cell aggregate within each microwell. And secondly, the agarose hydrogel has low adhesive properties. That is to say, it supports aggregate formation of non-adherent cells, as well as diffusion of cytokines and nutrients. And third, the conical microwells also allows determination of the filling level corresponding to the cell aggregate volume using microscopic or scanner imaging. And this is actually shown in the next slide. We call it the measuring cup principle, and we can here demonstrate the so-called measuring cup principle in T47D breast cancer and MIA-PACA2 pancreatic cancer cell lines, as well on the right hand side here MDA MB231, which is also a breast cancer cell line. The agarose microwell design allows spheroid formation in these cell lines, which typically are non-spheroid in other models. Also, we were able to show the feasibility of cell aggregate formation, here shown as volume distribution at defined cell numbers in MDA MB231, shown on the right hand side, and we can actually correlate that with corresponding scans of the disc-shaped device without actually harvesting the cells out of the device. If we need to harvest cells from CoSeedis, it's quite easy. The chip can be turned upside down, just flip it and centrifuge it so that the cell aggregates float into the media and then can be collected with a serological pipette. On the other hand, we can perform standard histology processing, and for this purpose, the chip is covered in 2% formalin solution. It can be sealed overnight with low melting point agarose, and we can do paraffin embedding and cast 2 micrometer thick sections for slide preparation, which can then be stained according to routine protocols, as shown here for MDA MB231 and for MIA-PACA2 with respective cell line specific marker profiles. If we now go back to our starting point and recapitulate why most models of myeloma have failed in predicting realistic drug efficacy at a later stage of clinical testing, we really need to check if our model is eligible for co-culture modeling. And the 2 classical approaches, contact co-culture and distance co-culture, were tested in this experiment using the T47D breast cancer cell line. And as you can see, both the contact and the distance co-culture setting could be established using CoSeedis. And on the right-hand side, you can actually see the readout, the volume expansion in T47D along with human MSC co-culture, and human MSC co-culture both benefited the growth of T47D breast cancer cell line under contact and distance co-culture settings. Moving on to myeloma, we first compared in vitro propagation of RPMI 8226 myeloma cells under 3 different in vitro co-culture settings, just culture settings at this point, sorry. These included a conventional 2D monolayer, a liquid overlay approach, and the 3D CoSeedis chip. For the later, we promote aggregate formation by an additional centrifugation step after seeding the cells, and indeed we observed proliferation kinetics that were quite different from the 2D and the 3D liquid overlay. Cell profilation in the monolayer and liquid overlay followed bell-shaped kinetics with a maximal growth at about day 6, followed by a decrease and full apoptosis at day 10, and cell proliferation in CoSeedis corresponded to a sigmoidal curve with an initial growth, delayed but more durable growth, which was ongoing after 12 days of observation. And to note, at day 12 also we checked the microscopical images of the different culture settings, and neither the monolayer nor the liquid overlay could actually foster cluster formation, whereas the cells within the microwells of the CoSeedis chip started to assemble and form instant cell aggregates within the conical microwells. Interestingly, when we move to co-culture experiments with human bone marrow stroma cells, here exemplified by the cell line HS5, both the ATP quantification and the transmitted light scanner technology revealed a slower proliferation rate for RPMI 8226 in the presence of HS5 as compared to monoculture, which from our understanding at least may be due to a delayed establishment of cell-cell communication by continuous cytokine secretion between myeloma cells and their corresponding niche partner. To test the permeability of the CoSeedis chip for cytokines and therapeutics, we transduced RPMI 8226 cells with the gene coding for the fluorescent protein M cherry, followed by subsequent incubation with cell trace CFCE and cell trace violet. Fluorescence was examined by confocal, and as you can observe in the lower panels on the left-hand side, both dyes permeated the cell membrane. Both dyes penetrated to the core of each cell aggregate, and shown here on the right, the agarose hydrogel itself did not absorb any of the fluorescent dye, thereby actually permitting a transparent background for imaging analysis. We next investigated the sensitivity of myeloma cells towards therapeutic agents within CoSeedis, and for this purpose we tested proteasome inhibition exemplified by the proteasome inhibitor bortezomib with well-established clinical efficacy against myeloma, FDA approved. And we tested a different class of agents exemplified by the D-ubiquitinase inhibitor uranafin with reported preclinical activity in multiple myeloma and other cancers. After 48 hours, drug-induced cytotoxicity in RPMI 8226 cells was substantially lower in the 3D model as compared to the normal 2D flat culture model shown for bortezomib on the left-hand side and for uranafin on the right-hand side. To note, sensitivity to both agents was even lower in the 3D model when HS5 cells were additionally provided as a co-culture partner. And as said, this observation was consistent across both classes of anti-myeloma agents. So we believe that we can conclude at this point that both the aggregate formation, so 2D versus CoSeedis, and the stroma co-culture foster resistance to anti-myeloma agents, which may be critical for a realistic approximation of later clinical efficacy to really detect and identify promising agents at the earliest possible stage of drug development. To evaluate the applicability of CoSeedis for ex vivo propagation of primary patient myeloma cells, we measured cytokine concentrations in the respective supernatants of RPMI 8226 cells, OPM2 cells, and primary patient cells derived from the bone marrow. And this was done using the Lunaris human multiplex kit. After 6 days of culture, we observed increased secretion of IL-1 beta, IL-2, IL-6, IL-8 and TNF-alpha in the presence of HS5 stroma cells. And other than expected, we observed a decrease of VEGF in the co-culture supernatants, which seems different to our current understanding on the role of neoangiogenesis and VEGF signaling for myeloma proliferation. It might be an issue of timing, so the next step currently ongoing is really to perform serial cytokine measurements over time, also by incorporating other cell types like endothelial cells, which may be even more relevant for stroma support in multiple myeloma to really individualize this model even further for the use of in plasma cell malignancies. Cell growth for primary patient cells was also assessed, and you've just seen the patient characteristics on the previous slide. So basically we tested primary myeloma cells from the bone marrow of 6 patients, all with advanced disease, some with relapsed refractory myeloma, some with initial diagnosis, but really a high tumor burden and high-risk cytogenetics. And we basically defined a limit threshold of at least 80% CD138, CD38 positive plasma cell infiltration in the bone marrow. And then we used these cells to perform growth monitoring over time, which revealed that primary patient cells actually do not expand in monoculture, even in the CoSeedis matrix. But if we add a co-culture partner, this picture changes. So human stroma cell line derived from a myeloma patient, MSP1, is quite beneficial for the ex vivo propagation of primary myeloma cells. We observe an about 2-fold increase in proliferation, but even more beneficial, HS5, the human stroma cell line you know already from our previous experiments, induced an about 6-fold increase in proliferation rate measured by ATP quantification, and the growth was still ongoing after 3 weeks of cultivation. And to really make sure that cells were viable, we performed multimodal assessment throughout the time at repetitive time points. And here you see the microscopy, Pappenheim staining, and IHC staining for CD38 in one patient after 14 days of cultivation ex vivo. To sum up, we believe that the 3D CoSeedis chip expands our current armamentarium for more realistic in vitro cultivation of cancer cells, and thanks to its aggro surface, it enables the cell aggregate formation in a scaffold-free system. It has this conical shape that can function, that can center the cells and can also function as a form of measuring cup principle, allowing for easy and accurate quantification of cell aggregate volume over time without the need to actually harvest the cells. The computer-aided design allows for high reproducibility and thus also allows for high throughput screening approaches. Importantly, monoculture experiments can easily be taken to the co-culture setting, both distance and contact co-culture. So this is very useful for cancers like myeloma, where we know that interaction with the bone marrow is crucial also for drug resistance. Which leads us to our specific conclusions in respect to multiple myeloma. In this part of our project, we observed that co-cultivation with human stroma cell lines within CoSeedis has a strong impact on the in vitro drug sensitivity across different drug classes, and it may be crucial for evaluating the efficacy of candidate drugs at the earliest stage possible. We could also observe that the survival and the proliferation of primary myeloma cells may be maintained with 3D CoSeedis up to a minimal cultivation period of at least 3 weeks, which seems unprecedented if compared to prior models. More work is needed and is currently ongoing to see how our model can be further adapted for the use in specific cancers and how it can synergize with other in vitro platforms. A lot of people have been involved in this project. I want to thank them on behalf of Andreas and me. I want to thank you for having us, and I'm sure Andreas will be very happy to answer all questions you may have. Thanks a lot.
Great. Thank you so much, Dr. Waldschmidt, for your presentation. And before we begin the Q&A session, I want to make sure that you look into the chat feature. I did post some ways to contact Dr. Waldschmidt. And it looks like we have a few questions. And I'm also going to post, I'm looking at the paper where you can get more information, and I will also post that in the chat as well. So if I can get Dr. Marco Lowe and Dr. Andreas Thomsen to rejoin me. And we're going to get started with the Q&A session. I just want to post this link for everyone in the chat. And before we begin, let me just indicate we have 2 questions in the Q&A so that we can better manage the questions. Please submit your questions in the Q&A feature. Thank you so very much. So we'll go ahead and begin, and looks like we have questions in the Q&A, and I'll go ahead and get started. The first one is from Giovanna Armentano. He says, "Hi everyone, I'm a Brazilian master student, and it is the first time I'm hearing from 3D cell culture. I work with a syndrome called cardiorenal, which I do a hypoxia in renal cells and then transfer the medium from cardiac cells to study if cytosines can do a crosswalk within the organs. Do you think I can use a 3D co-culture with both lineages as a method? How much does it cost?"
Okay, so this is quite an interesting question, Giovanna. Thank you so much. I think this question you are trying to answer on cardiorenal syndrome could be answered by a 3D co-culture model. You need to, at first, you need to find out if the cells you're using are able to grow in 3D. So there's a number of cells which grow very nicely and a few cell types which need some anchorage dependent conditions. So this would be the first step to find out. If one of the 2 cell types you're using is working well in 3D, you could seed these cells into the conical cavities and have the other cells underneath the chip as a partner where they can adhere to the bottom of the plate. And the communication will take place through the agarose gel, so that cytokines and other messages can be exchanged in a distant co-culture way.
Okay, thank you for that. Terry Risk wants to know, have you done any direct comparisons to Corning ultra-low binding plates?
I've been working with these cells, with these plates from Corning. They're working nicely. The point which drove me away from these plates is that the aggregate formation is not controlled in any way. So if you have one plate, you will have 100 or even more aggregates of different sizes. And so this was one reason why we came up with this micro-patterned or micro-cavity array, where we have a control on the number of cells and we have a control of the size of the aggregates which are formed. Overall, I can say that those cells which will grow in ultra low attachment plate will definitely also thrive in such a microcavity assay.
Maybe I can add to that as well, Andreas. One reason, of course, which drove us initially to develop 3D CoSeedis was also that we tried to establish rather more complex systems in which we had different cell types and different organoids or spheroids communicate with each other, either in contact or in distance. And that is, of course, something which is more difficult to or not achievable in certain Corning plates. And as well, one aspect that we also had is that for some assay, it was absolutely required that we could keep the cells in culture for really long periods of time. And I'm speaking weeks here, which again, there are some problems with plastic plates in general, which normally do not maintain long cultures too well. So that drove us to really develop a system whereby, such as CoSeedis at the end.
Okay, thank you so much. And please forgive me if I mispronounced your name. And next we have a question from Sabine Mai. Two questions. One, how about antibody or biospecifics in 3D culture of MM? And the second question is, have you tried bone marrow aspirates in 3D culture?
Okay. So I will start. So I will start with the second question, which is more quickly to be answered. We did not seed bone marrow aspirates into the microcavities so far, but we did it with hematopoietic stem cells from umbilical cord blood, and they formed colonies very much like in material cellulose. So after all, we could use this array could be used as a standardized substrate for hematopoietic stem cells. So this was the second question. And the first question I will point over to Marco, because he did some work not in myeloma cells but in other cell types and antibody conjugates.
Yes, exactly. I mean, one of our big projects is in fact to test antibody drug conjugates in 3D CoSeedis on either microtumors, for example, but also liver or kidney organoids. And all I can say is that we have achieved very excellent results. Antibodies freely diffuse to the organoids. They elicit their function there as expected. And the nice thing is we can really do what we call in-chip assays to monitor cytotoxicity, cell viability, cell growth, all in a relatively easy and a really statistically relevant fashion, because we can constantly measure the effect of a certain antibody on hundreds of microtumors simultaneously. And these microtumors, they all grown under the very same conditions and were exposed to the very same concentrations of antibodies. So they faced the same environment all over the assay, which of course makes the data extremely relevant at the end.
Okay, and anything else to contribute on the first question? Everything's answered, right? Okay, Kabita Sharma has a question. Is it enough to use formaldehyde to co-culture the 3D organoid model cells while subculturing from the scaffold of the cells to avoid contact inhibition, specifically if the model is solid tumors? Andreas, would you like to start?
I must state that I would not use formaldehyde as long as the cells are viable. So we use formaldehyde to fixate the cells and the aggregates prior to paraffin embedding. But I must say that this question needs some explanation.
Okay, anything that you would like to add, Dr. Lowe?
Well, all I can say is that we have observed in the past that all conditions that were previously established in either 2D or in other 3D systems can be easily transferred into the 3D CoSeedis chip as well. I don't see why this shouldn't work really. If you have worked out concentrations as little to avoid contact inhibition, then that should work as well. I would be sure that could be tested quite easily as well.
Okay. Well, we have one additional question. It's in the chat, but I'm going to go ahead and read it. It's from Abbas Fazal. And Abbas says, I really enjoyed the presentation. By using 3D bioprinters, it's feasible to incorporate the cells. Why do you believe that it's complex? Would you please tell us the benefits of 3D bioprinters for disease modeling? Thanks a lot.
Okay, so bioprinting is a really interesting and yes, it's a very promising point to assemble cells and extracellular matrix into some construct. At the moment, all approaches which I know are not able yet to form a real tissue-like structure so far. So at the moment, my recommendation would to just keep on working on this, and otherwise find out what interactions can be found if you seed different cell types together into such a environment like the 3D CoSeedis or another 3D cell culture model.
And
Marco,
do we have
any addition?
No, I completely agree. I mean, there are certain applications for which 3D cell modeling, printing is certainly a very nice feature. However, in the context of 3D CoSeedis, we never really came across 3D printing as such. Since the system of 3D CoSeedis works in a different setup. But of course, for reconstituting, for example, certain organs or extremities such as nose or ears, 3D printing is certainly a promising technology.
Well, Abbas Fazal has one more question. I'm curious to know the concentration of your agarose gel.
Yes, I can quickly answer to this. So the concentration of agarose is higher than it's usually used because we need a certain mechanical stability. So it's usually 2.4%.
Okay. Okay, well, we have two more questions. And one says, you showed that you could maintain primary myeloma cultures for up to 3 weeks. This is indeed very long. What do you think is the role of long-term cultures in preclinical models?
Mm-hmm. Yes, I can really confirm that the usual 3 cell culture models are really extremely short-lived. So this is a huge contrast to what happens in vivo, and we definitely see a lot of things happening not only within the first few days, but we had, for example, something like tumor recurrences after 5 to 6 weeks in our 3D model. So these recurrences, these actions would have been completely missed in a short-term culture. So from this side, it makes a lot of sense to look at the longer term and to have models which allow such a long cultivation period. Marco, do you think you could add anything?
Yes, well, I would just like to emphasize that. I mean, we developed the 3D CoSeedis long-term tumor relapse assay in this context, which, to my knowledge, is the only assay on the market that addresses this question. So it actually addresses long-term drug efficacy. Because, as you certainly agree, Andreas, it has been or is still an issue in clinics that you not only want to kill off proliferating tumor cells, for example, but you want to actually kill off quiescent cells as well that eventually otherwise repopulate a tumor. And so far, to test that in ex vivo has been impossible. And there are two reasons for that. First of all, these are rather stochastic events. So you need to have very large numbers of tumors that you can look at in order to pick up those few that do eventually survive. But of course, in clinics, these are important results. One cell is enough eventually to regrow the tumor. And the other issue is exactly that you need to, sometimes you need to grow those tumors over weeks to see that. And so you need a system in which you can keep those microtumors alive over weeks, and that is an issue with a lot of systems, and I think we solved or present a nice solution to that problem there.
Okay, and we have one more question. What is your experience on serum reduced or even serum free 3D culture?
Mm-hmm. Yes, this is also quite a good point. It's because that we found that, or others have also found that as soon as you move into a system where the density of cells and the amount of cell-derived factors increases, and the cells have an ability to create their own microenvironment, we need much less serum than we are used to use in monolayer cultures. So in a number of breast cancer cell lines, we have virtually reduced serum to almost 0% and still keep the cells in a very good condition for very long periods of time. And we definitely must state that FBS is a factor which is highly unphysiological. If you use it in your culture, you usually need to use it, but to get rid of this 3D culture overall is a very nice opportunity.
Okay, well, you know, everyone's really engaged and excited about this. I have two more questions that come in, and I do believe we have time for them. So Terry Riss wants to continue her his or her question. Where you showed n equals 4 on the graph, was that 4 wells each with multiple spheroids or just 4 spheroids?
Okay, I have to move to this graph. It was always a cohort of series, so it was some larger number of aggregates which was followed at the same time.
Okay. And Kavita Sharma she wants to continue her question. And if the cells are to be harnessed for FFPE sample preparation for spatial phenotype analysis, while administering the MABS-based therapeutics regime, is it necessary to consider only a particular set of the tumor organoid model if he misses out of therapeutic activity due to co-culture heterogeneity? Should I repeat the question?
Yes.
Andreas?
The first one. Marco, do you have an instant answer?
Well, if I understood the question correct, it is whether the spheroids need to be harvested in order to embed them for paraffin sectioning or paraffin embedding. And the answer to that is no, we can do everything in chip again. So actually, The nice thing is we don't need to handle individual spheroids, so we don't manipulate them individually, we don't damage them, we don't lose them. Everything is done in chip, dehydration, as well as paraffin embedding, and then you can section through the chip and the spheroids at the same time, and you kind of end up with a sort of like a microtissue array on your slide, which makes it very easy to do immunohistochemistry later on then.
Okay. Did you want to add anything, Dr.
No,
Thomsen?
I think this is the correct answer to this question.
Okay, great. Well, that's all of the questions that we have for now. And I want to thank everyone for your questions, your comments, your participation. But before we conclude, we have an additional poll question for you. We want to find out, we'll post the poll now, how many of you will use 3D cell culture now that you've heard this presentation? Great. Okay, what do you think about those numbers?
Wonderful. So 3D culture is a step ahead, and everybody who's joining this community is very welcome.
Great. What about you, Dr. Lowe?
No, great. I mean, I think at the end of the day, it's great that we move into more complex biological systems. Also, of course, we always have in mind to replace animal models as much as we can. Not only is it quicker and cheaper, but it's also kind of a moral obligation to really do as much as we can ex vivo. And I'm glad we move in the right direction there.
Okay, and it looks like in the last question, Capita is indicating, thank you for clarifying. So you did answer the question for understanding there, and we're getting some good feedback about how much they've enjoyed this presentation. So if we can just get everybody back on video and audio, we'd like to just thank you very much for giving this wonderful, exciting presentation on your technology, Dr. Thomsen, Dr. Lowe, and Dr. Waldschmidt. We thank you so much. Do you want to make any closing remarks before we end?
Thank you for listening. It was a great pleasure.
Thanks again. And I'm looking forward to interesting follow-up questions.
Yes, and we have Serge. Since Serge, you came onto the camera, why don't you introduce yourself and let us know who you are?
Yes, hello everyone. I'm Serge Kadura with Nanoscale Labs. I'm the US partner of ABC Bioplay. So I'll be getting in touch with you if you need help and if you're in the US. Thank you very much.
Okay, and I also would like to have Jacob White get on video. I'd like to thank him for inviting me to moderate this incredible webinar and all that he does behind the scenes to make things happen. So thank you so much, Jacob. All right, and thank you to all of you. Again, you have all of the information in the chat. You can feel free to save the chat where you can contact Dr. Waldschmidt. And of course, if you have any questions, you can definitely reach out via email. And we'll have this available on demand within 24 hours on our APS website. Thank you so much for joining us and have a great day.
Tricia, thank you so much for
Thank
this
you.
nice
Bye-bye,
invitation.
everyone.
Okay, bye-bye.
Bye.
Bye.